Welcome back to Channel 1919 Westwood Daily News. I'm your host, Aiden Torres, and today we have a special report about a breakthrough in science. Joined here with me today are two student researchers, Thibaut Lucet and Patrick Villanueva, here to talk about a game-changing innovation in the battle against antibiotic resistance, phage therapy. Welcome, gentlemen.
Thanks for having us, Aiden. We're excited to finally report this new innovation that I feel most people don't know about.
Right? It's basically like nature's own microscopic hitman, taking out bacteria with surgical precision.
Wow, that sounds intense, but let's back up just a little bit. What exactly is phage therapy?
That's a great question, Aiden. Phage therapy uses bacteriophages, viruses that infect and kill bacteria, to treat bacterial infections. Unlike antibiotics, which target a multitude of elements in your microbiome, phages are highly specific and only target harmful bacteria.
They are designed for precision and accuracy. Instead of wiping out everything, including good bacteria, phages only attack specific bacteria. And the best part, they can evolve alongside bacteria, making it much harder for a resistance to develop.
That sounds like the perfect alternative to antibiotics. But if it's so great, why aren't we already using it?
Well, we actually were. Phage therapy was first discovered in 1917 by French-Canadian scientist Félix Sterel. By 1919, it was successfully used to treat a gastrointestinal disease.
But then came antibiotics. The West got distracted by penicillin in the 1940s and phage therapy got sidelined. Meanwhile, the Soviet Union, and especially in Georgia, kept developing it in secret.
Classic Cold War plot twist. But now, with antibiotic resistance on the rise, phage therapy is making a comeback?
You got it. Scientists are now engineering phages to be even more effective and exploring ways to integrate them with antibiotics.
Okay, so let's get into the nitty-gritty. How does phage therapy actually work?
Well, Aiden, there are a few different approaches. The simplest is the lytic cycle, where a phage attaches to a bacterium, injects its genetic material, hijacks the bacterium's machinery to create more phages, and then boom, the bacterium bursts like an overinflated balloon.
There's also sequential phage therapy, where if a bacterium develops resistance, we introduce a new phage, a phage that specifically targets the resistant strain in a different way.
Wow, sounds like bacteria are really playing a losing game here.
Pretty much. And to make phage therapy even stronger, we use phage-derived enzymes, special proteins that break down bacterial defenses. These include B polymerases, which dismantle protected bacterial proteins, and endolysins, which basically punch holes in bacteria from the inside. Think of them as biochemical wrecking balls, smashing through bacterial defenses to clear the way for disruption.
There are also virion-associated peptidoglycan hydrolases, a mouthful, I know, but they help break down bacterial cell walls, making it easier for phages to infect. And then we have polyps, which create tiny holes in bacterial membranes to allow endolysins to finish the job.
So we're not just using phages themselves, we're also using their weapons.
Spot on. And these enzymes can be used alongside phages or even as standalone treatments.
That's incredible, but I hear researchers are going even further with bioengineering.
Absolutely. Scientists are developing CRISPR-equipped phages that can edit bacterial DNA and eliminate antibiotic-resistant genes.
And let's not forget chimeric phages, custom-built hybrids designed to be more efficient and versatile against bacteria.
So we're basically weaponizing viruses against bad bacteria. Wow, science is wild.
It really is. But it's not just about killing bacteria. It's also about enhancing existing treatments. And this is where immunophage antibiotic synergy comes in, combining phages, antibiotics, and the immune system for a stronger attack.
So how does that kind of work?
It is actually quite simple. When a bacteria mutates to resist phages, they often become more vulnerable to antibiotics, especially if phages target their efflux pumps. Plus, when phages burst open, bacteria, the fragments activate the immune system, making it more aggressive against the infection.
So phages aren't just killing bacteria, they're boosting antibiotic effectiveness and activating the immune system at the same time.
Exactly. And that's why phage therapy is seen as a complement to antibiotics, potentially restoring the effectiveness of older drugs.
Wow, that's a game changer. And what about bioengineered phages?
Scientists are designing chimeric phages that combine the best traits of different phages, allowing them to infect multiple bacterial strains instead of just one.
And another big breakthrough is phages as drug carriers. They can be engineered to deliver antibiotics or enzymes directly inside bacteria, bypassing defense mechanisms like efflux pumps.
And the most mind-blowing part, some phages can actually trick bacteria into self-destruction by hijacking their own CRISPR immune system, forcing them to attack their own DNA.
We're making bacteria turn against themselves? That's both terrifying and brilliant.
It is. And with CRISPR phages, immune synergy, and bioengineering, we're moving towards highly precise infection treatments.
Instead of broad-spectrum antibiotics, phages offer a way to target only the harmful bacteria, leaving the beneficial ones untouched.
And as research continues, phage therapy could become a mainstream tool in the fight against antibiotic resistance.
That's both fascinating and reassuring. It sounds like phages are the future of infection treatments. So, where is phage therapy actually being used today?
Well, one area where phages are showing huge potential is in the treatment of UTIs.
Right? In fact, a 2025 study demonstrated that phage cocktails can effectively treat UTIs caused by antibiotic-resistant E. coli and Klebsiella pneumonia.
And the results are promising. Phage therapy not only successfully eliminated these bacteria in clinical trials, but it also reduced inflammation and improved patient outcomes, even in cases where antibiotics had completely failed.
That's huge. So phage therapy is already making an impact in medicine. What about tuberculosis?
That's a great question. Another study explored the use of macrobacteriophages, which are phages that specifically target Mycobacterium tuberculosis, the bacterium responsible for TB.
And TB is a major global health concern, especially with antibiotic-resistant strain emerging. But this study found that phage therapy could effectively lyse M. tuberculosis without harming human cells. Early-stage clinical trials suggest that phages could actually be a powerful alternative for treating drug-resistant TB.
So phage therapy is being tested against some of the most dangerous antibiotic-resistant infections, and it's actually working.
Yes, and beyond medicine, phages are also being explored for food, safety, agriculture, and even environmental sanitation, all areas where bacteria are a growing problem.
So we've talked about how phage therapy is transforming medicine, but I hear it's also being used in other industries. Is this correct?
That's right. Phages aren't just for treating infections. They're also being explored in food safety, agriculture, and environmental sustainability.
Take the food industry. Phages are being used to eliminate foodborne pathogens like Salmonella and E. coli on food products and processing surfaces. Since they only target harmful bacteria, they act as natural preservatives without harming beneficial microbes.
So we can make food safer without relying on chemical preservatives or antibiotics.
Exactly. And that same concept applies to agriculture. Farmers are currently using phages as biocontrol agents to combat bacterial plant diseases. Unlike chemical pesticides, phages provide an eco-friendly alternative without harming the environment.
Phages are not being studied for pest control, not by killing insects, but by targeting the bacteria that some pests rely on. This helps reduce pest populations without affecting beneficial insects or using toxic chemicals.
That can make agriculture much more sustainable. What about environmental cleanup?
Good you bring it up. Researchers are actually testing phages for water treatment right now, where they help reduce bacterial contamination and improve water quality.
And in aquaculture, phages are used to control bacterial infection in fish and shellfish, reducing the need for antibiotics in fish farming and making seafood production safer and more sustainable.
I never would have thought of phages in fisheries or water treatment, but it makes total sense.
And actually, one more big one, environmental sanitation. Phages are being used in wastewater treatment and soil cleanup to target harmful bacteria, making them a promising tool for bioremediation.
Wow. So phages aren't just revolutioning medicine. They're also improving food safety, agriculture, pest control, water treatment, aquaculture, and environmental cleanup.
Exactly. They're proven to be an incredibly versatile tool, tackling bacterial problems without disrupting the ecosystems.
And as research continues, phages can become a key player in sustainability and bacterial control across multiple industries.
That's amazing. It really sounds like phages are nature's secret weapon, not just against fighting infections, but across so many different fields. This all sounds incredibly promising, but there must be some kind of hurdles or something to overcome, right?
Absolutely, Aiden. While phage therapy offers a targeted approach against antibiotic-resistant bacteria, its journey from bench to bedside is actually met with significant challenges. One major hurdle being regulatory approval. Unlike standard antibiotics, phages are dynamic, self-amplifying agents. They require personalized cocktails tailored to each patient's specific infection, or a one virus per target approach that complicates both standardization and testing, ultimately delaying broader clinical usage.
And on top of that, there's the issue of production and scalability. Traditional phage therapy involves isolating, purifying, and amplifying the correct virus, often under extreme time pressure when patients are critically ill. Without rapid purification methods, even a well-designed therapy might arrive too late. Plus, the need for customized phage cocktails means the production process is both labor-intensive and costly.
Wow, that sounds like a high-stakes race against time and regulatory red tape.
Exactly. But here's where the silver lining comes in. These very challenges are driving innovation. Advances in genetic engineering and synthetic biology are actually opening the door to designer phages, viruses engineered to overcome bacterial defenses more efficiently. For example, by combining multiple phages that target different bacterial receptors, we can drastically reduce the chance of resistance emerging. This idea, reminiscent of the Red Queen dynamics we discussed in class, forces bacteria into an evolutionary corner.
Plus, there's exciting progress in automating the process. Automated platforms and high-throughput systems for rapid phage identification and production are on the horizon, which could transform our current laborious methods. Researchers are also exploring enzymatic approaches to break down biofilms and are investigating strategic integrations of phages with antibiotics. In the future, phage therapy might not only serve as a standalone treatment, but also as a crucial component of a sophisticated multi-pronged attack against complex polymicrobial infections.
So, while there are significant challenges, these innovations could eventually make phage therapy a game-changing solution in our fight against resistant bacteria. Is this correct?
Exactly. Every obstacle we face is also an opportunity for breakthrough advancements. It's an exciting time for this field, and we're really just scratching the surface of what is truly possible.
Wow. Thanks for that deep dive into the challenges and opportunities of phage therapy. It's clear that while the path forward is complex, the potential rewards are enormous. As we conclude our journey through phage therapy, let's now recap our key insights. We discovered how bacteriophages can selectively target harmful bacteria while sparing the beneficial ones, a method that's evolving right alongside our challenges with antibiotic resistance.
From its early days to a period of decline and now a modern revival, phage therapy has advanced through both conventional methods like the lytic cycle and phage cocktails and cutting-edge strategies such as designer phages and immunophage antibiotic synergy.
Although hurdles like personalized treatments and labor-intensive production remain, these challenges are sparking innovation. Advances in genetic engineering and high-throughput production are paving the way for a multi-pronged attack against infection.
Well, that's all the time we have today. A huge thank you to Thibault Lucet and Patrick Villanueva for joining us on the show today. Until next time, stay curious, stay informed, and wash your hands. I'm Aiden Soares, and this is Channel 1919 Westwood Daily News.